Before we even begin to talk about meiosis, we have to remember where this process and where mitosis are occurring in your body. So mitosis is occurring pretty much everywhere, right? There are a few cell types that are stalled out in interphase like neurons, but almost all other cells in your body do mitosis, almost. And so these cells that do mitosis are called somatic cells. In contrast, what are generally called sex cells are contained in ovaries and testes, can be passed on to offspring, and do meiosis to create the gametes, or sperm and egg, involved in sexual reproduction. So it's important to realize that you can't pass on mutations in somatic cells. Only cells that do meiosis contribute to the next generation. I do kind of love this life cycle picture here. I feel like it has a lot of vocab that you need to know for meiosis, and that it gives a nice visual that helps students to remember some of the important things about meiosis. So let's start with the key down at the bottom right of the picture. Let's get in our minds the background colors of beige and blue. Beige is going to represent cells that are haploid or have only 23 chromosomes in them. Blue is going to represent a term called diploid, and that will represent cells that have 46 chromosomes in them. So let's start with a diploid structure, U. Your somatic cells are diploid with 46 chromosomes. The cells that perform meiosis are contained in the ovaries and testes. Stem cells in your ovaries and or testes complete meiosis to produce what are called gametes, or egg cells and sperm cells. These gametes are haploid, and they only have 23 chromosomes because meiosis reduces the numbers of chromosomes by half. That's an official definition of meiosis, so don't forget it. Now that we have our haploid gametes, they're going to come together in a process known as fertilization. And then the resulting cell of that is called a zygote, and it's diploid because 23 plus 23 equals 46. This zygote then divides by mitosis, making more and more copies until you get to the structure that you are today. And since the zygotes divide by way of mitosis, making clones, all your cells have the same 46 chromosomes. Sometimes it's helpful to look at a karyotype, which is the picture on the left here. This is just a picture of all 46 chromosomes laid out by size. Remember, chromosomes come in pairs. You get one from mom and you got one from dad. So this picture clears up some of the visual aspects of your chromosomes and how they interact in inheritance. Don't forget this picture. You have 46 chromosomes, but that's like saying that you own 46 socks. Now, if you're like my husband, that's no big deal because you buy your socks in bulk, like once a year, and they actually are all the same. But let's just say, for example, I have 46 socks, and they are in pairs. And let's pretend that my socks have toes on them, and they come in pairs with a left and a right specifically designated. Your chromosomes come in pairs, and they're specifically designated. You get one from mom and one from dad. Now, let's apply that to the karyotype on the left again. Which one in each pair is from mom and which one is from dad? You can't kind of tell. Well, you can't tell from just the picture. But the fact is that you have 23 pairs of chromosomes. And you can sense how you got 23 of them from sperm and 23 of them from egg for your zygote. Think about for a minute how you might make your own gametes, giving away only 23 of these chromosomes, one from each pair. I also like this picture of meiosis. I like it because you can read it from left to right, but I also like it because it starts with a pair of homologous chromosomes in their unduplicated state. And this right here is usually a point of confusion for many students because many books will start with that second cell right there with the chromosomes already in their duplicated state. So I have one big thing to say for you to remember. To know how many chromosomes are in a cell, you need to count the centromeres. Check out how the first circle on the left has 2 centromeres. The next circle to the right of it in the sequence, it also has 2 centromeres. However, the 2 chromosomes are in their duplicated state with sister chromatids. Meiosis 1 is going to separate the homologous chromosomes, or the matching pair, one from mom, one from dad. Meiosis 2 is going to separate the sister chromatids, very much like mitosis. We know this is meiosis, this picture here, because meiosis reduces the number of chromosomes by half, and the beginning cell has 2 chromosomes, and then our ending cells each have 1 chromosome. This is a great visual of the difference between meiosis 1 and meiosis 2. Meiosis 1 is separating the homologous chromosomes, or the mom-dad pairs, the left-right socks. The pictures on the right are separating the sister chromatids by splitting the centromeres, and that's much like mitosis. I've added in the next 2 slides after this just to provide the process of meiosis 1 and 2. There are so many videos online. I strongly suggest watching the process of meiosis as a video. It's much more meaningful to see it in motion. Both meiosis 1 and meiosis 2 proceed with the same 4 phases each, and the same details, like things like the nuclear envelope disappearing and reappearing, and the formation of the spindle. So if these details are fuzzy to you, I would strongly suggest going back to the mitosis mini lecture instead of trying to digest them here. We learned about those details with our learning about mitosis, and questions on the quiz will ask about those details against the background of mitosis. So what am I going to ask in the questions about meiosis? I will definitely ask about the splitting of the chromosomes, focusing on the metaphase states. This really is the key to understanding meiosis. So let's focus here on the third picture from the left in metaphase 1 of meiosis 1. Note 2 things that are different than a metaphase of mitosis. Number 1, the chromosomes, they're not all one color. It seems like the tips of them have been exchanged with their partners. All right, noted. Number 2, the chromosomes are aligned along the metaphase plate by their pairs, whereas in mitosis, each one lay along the metaphase plate. And this reflects the statement at the top of the slide: meiosis 1 separates homologous chromosomes. These are the pairs of chromosomes that you have. Think about the karyotype, separating those pairs. Now that we've separated the homologous chromosomes, let's move on to meiosis 2. You only have to follow one level on this diagram, the top or the bottom, to get the understanding of meiosis 2. So let's focus in on metaphase 2 picture, the third from the left. These look like mitosis. We're breaking apart those sister chromatids. But these cells, these have 3 chromosomes, which is half that of the original cell. Yes, this is the reality of meiosis 1. Here in meiosis 2, we're just returning the chromosomes to their original non-duplicated state. Meiosis 2 ends with cells with half the number of chromosomes. And we also end with 4 cells as a result of the 2 rounds of division. Independent assortment is something that happens in meiosis 1. It is a source of genetic diversity that can create over 8 million variations in one human gamete. This figure here is based on 2 sets of 23 chromosomes. This picture here gives you a great visual of independent assortment. Check out the cells at the top of each picture, which each of them has 2 pairs of chromosomes in metaphase of meiosis 1. Note how there are 2 possibilities for this cell as to how the homologous chromosomes line up on the metaphase plate and separate. Right? All blue on one side, all red on one side, and then possibility B has blue and red alternating. Now extend this to think about how our 23 pairs of chromosomes can split up. Not 2 pairs, but 23. The key to understanding independent assortment is that 1 pair of homologous chromosomes lines up on the plate without any regard to the other pairs. And this creates 2 to the 23rd possibilities, or 8 million different kinds of gametes that you can create. But we don't stop at 8 million combinations. It's actually closer to infinite due to a process called crossing over. This is a process that also happens in meiosis 1. When the homologous chromosomes pair up in prophase 1, they exchange a little bit of DNA with each other. So your mom chromosome and your dad chromosome, they switch some DNA. Take a look at this picture here. We call chromosomes in their duplicated state, we call them tetrads. So tetrads are made up of sister chromatids for 1 chromosome. The tetrads have a leg or an arm that touches the tetrad of their homologous pair. And this touching site is called a chiasma. It's at this site that the exchange takes place. So look at the resulting 4 chromosomes that will now be in the gamete. If you think about it, these chromosomes are completely new to the human gene pool, never before seen. The combination of genes on these chromosomes is completely unique. Crossing over is sometimes called genetic recombination, and there can be more than 1 chiasma during crossing over, creating combinations like the ones shown in this picture here. Crossing over also breaks linked genes, which are genes that are normally found on the same chromosome and are commonly inherited together. So as a very, very general example, we know of certain pigmentation combinations that are common. We have the light-skinned, red-haired, green-eyed Irish combo, and we have the olive-skinned, dark-haired, and dark-eyed Mediterranean combo. There are many examples of the offspring of these 2 phenotypes, one being the often present, auburn-haired, dark-eyed, but light-skinned variant that's present in Ireland.